Hello everyone, welcome back to the fourth and final section of special senses. Here we're going to be talking about equilibrium and hearing. So these are two closely associated senses that share a common mechanism, even though you might not normally think of them being as connected, right? Your sense of balance and your sense of hearing seem to be totally unrelated, but they are actually very closely related and they use the same type of mechanism to function. So this is by far and away the most complicated and confusing section of special senses. So I would really strongly encourage you, either before or after listening to this lecture, definitely read the section on the book. It will really help clarify. There's a lot of confusing terminology, a lot of terms overlap, but you need to keep them as separate and distinct. So I would strongly recommend also having a little notepad and writing out definitions and make sure you have them very clear in your head in order to avoid confusion. Okay, that being said, let's dive right in. So your sensation of equilibrium or balance and hearing are both stored inside your ear, right? So the ear has three distinct regions going lateral to medial, external, middle, and inner ear. So you can see in this image, most laterally, we're going to have the external ear, which is made up of the auricle, which is the actual fleshy part of your ear sticking on the side of your head. That's your auricle. And your internal acoustic meatus, that's your ear hole, your outer ear hole, your external acoustic meatus. This will end at the tympanic membrane. That's just a fancy word for your eardrum. So the boundary of the external ear is from your auricle to the tympanic membrane. Now, deep to the tympanic membrane, you're going to get into the middle ear. So this is an air-filled space. So you're going to have this cavity right here. It's called the tympanic cavity. And this is where you're going to find your three auditory ossicles, or the three ear bones, the Malleus, the incus, and the stapes. Malleus meaning like mallet, I mean it's a hammer. Incus means it's an anvil, and stapes is a stirrup because it looks like a stirrup that you put your foot in when riding a horse. So the middle ear is going to end at this structure here called the oval window, which the stapes is sitting on top of. Now the tympanic cavity, this air-filled space where you'll find your three bones, is actually going to be connected to the nasopharynx by a structure called the auditory tube. This is sometimes known as, the older name for this is the eustachian tube. So if anyone has been on an airplane or driven over the mountains in a car, your ears pop and you swallow to get your ears to pop so your ears stop hurting, that's what's going on here. So when you yawn or open your mouth or swallow, what you're trying to do is open up that eustachian tube. So if you're in a plane, you're at sea level and the plane takes off, you're going to be at a much higher altitude, which means there's less air pressure. So what that means is inside your ear, there's going to be a very high pressure relative to the outside air, which is at low pressure. This will cause your tympanic membrane to be pushed, and that will be uncomfortable. The way you avoid this popping or bursting is to equalize the pressure through this tube, through the auditory tube. When you yawn, your ears will pop, and then you'll have the same pressure from the mouth to the auditory tube to the tympanic cavity will be the same as the outside, and then you're going to feel relief. Everything's going to be just fine. Now, what often happens with ear infections, if this tube gets swollen shut because of inflammation, fluid can build up in this cavity, and then the fluid starts pressuring your tympanic membrane, and that really, really hurts. And one of the reasons why kids are so much more likely to get ear infections is because these auditory tubes, or these eustachian tubes, in kids are really, really small, and kids get lots of colds, so it's going to get sick. These little channels are going to close off, and it's going to fill with fluid, and that's how your little one ends up with so many ear infections. Okay, now that takes care of the external ear, the middle ear. Now the inner ear. This structure here, made up of the vestibule and the cochlea, like concha, meaning shell, this is inside the temporal bone. So remember back to the skull, we had that petrous portion of the temporal bone, where this stuff is inside the temporal bone. Here's where you're going to have the various structures for hearing down in the cochlea and equilibrium in the vestibule, and these things here called the semicircular canals. So here you're going to have receptors that are going to take the stimuli of sound and relative position, turn those into action potentials, and then that will travel up your vestibular cochlear nerve to your brain. So external ear, I already covered these things. Right, this thing right here is your auricle. This ear hole is your external acoustic meatus, which ends in the tympanic membrane. Sound is just compression waves of air molecules, causes your tympanic membrane to vibrate, causes your auditory ossicles to vibrate, which then allows your inner ear to process the sound. Now, we have just a little slight side bit here. Inside your ear, you can get earwax. What earwax is a compound called cerumen, which is very similar to what sebaceous glands produce, but here the sebaceous glands are a little bit modified, and they make something that's a little more solid, a little more waxy, a little less oily. Okay, middle ear, right? That's where we have this air-filled cavity called the tympanic cavity. And where you're going to have your eustachian tube or your auditory tube connects to your nasopharynx. So that's part of your respiratory system. So it's going to connect to your throat. And here we have the three bones of the middle ear, the hammer, the incus, and the stapes. If you want to call it malleus, incus, and stapes, great. If you want to call it hammer, anvil, and stirrup, just fine. Both those sets are acceptable. One thing I do want you to know is to know that the hammer of the malleus is lateral, the incus is more medial, and the stapes is most medial on either side. So I do want you to know which is lateral and which is medial. So the malleus connects from the tympanic membrane to the incus connects the malleus to the stapes, and the stapes is going to connect the incus to the oval window, which is part of the cochlea. So here is our image. Here is our external acoustic meatus. This is the tympanic membrane connected to the hammer, connected to the anvil, connected to the stirrup. The stirrup is then connected to the cochlea through the oval window. So do you guys remember what the longest muscle is in the human body? All right, that was the sartorius big. Here in the ear, we have the shortest skeletal muscle in the body. And this is little tiny guy right here, the Stapedius. The Stapedius is the shortest skeletal muscle in the human body. So along with this guy here, right, this is called tensor tympani. Now, if you look at these muscles, you might be asking yourself, why do we have muscles in the bones of our ear? Right? The purpose of these bones was to take the vibration from the air, a vibration from the air, and direct that into the inner ear. Well, these bones actually do more than simply transfer the energy. They actually magnify the energy. In fact, these bones serve as a system of levers that magnify that vibration by a factor of 20 or 30, depending on who you want to quote. So this is very important for sound amplification. And that actually gives you a hint to why we have these muscles here. And the reason is, if you are going to experience a loud sound over a long period of time, what will happen is these muscles will contract. These muscles will clamp down and prevent these bones from vibrating. If they're not vibrating, they're not magnifying. If they're not magnifying, they're actually protecting your inner ear from damage. So if you're exposed to a loud sound for a long period of time, yes, it definitely does cause hearing damage. However, it is not nearly as bad as a sudden, unexpected loud sound. So one of the ways we're actually able to work this out is, believe it or not, some of the people that have the worst hearing damage in the world, by population or region, are the people that live in the quietest part of the world. So you can say to yourself, Why would people living in extreme silence have hearing damage? Well, when they looked into it, they found it was in a group of Inuit people, so Eskimo, but people who live in the Arctic Circle. And it was only in adult males. It wasn't a problem anywhere else. And the reason is adult males in this population were the only people engaged in seal hunting. And how you hunt seal is you go out onto the ice floes, you find an open hole, and you just sit there with your rifle and wait for a seal to pop out. So they're in the quietest part of the world, waiting in silence for the seal to show up, and then they pull the trigger, which of course, bang, is a loud noise, and there's simply not enough time for these muscles to tighten down and protect the ear. So you get this massive shock wave of sound that basically has a lot of jarring motion, and it ends up damaging the inner ear. And because it happens so quickly, there's no time for these muscles to protect the ear. Whereas other people who are working construction or military or whatever, people exposed to really loud sounds, yeah, it's not good. But if it's going on for a period of time, your body has a chance to react and actually protect your hearing. So, little fun fact for you though, if you're going into a place where you expect sudden loud sounds, definitely wear hearing protection, because it's way more dangerous, because your body's normal way of protecting itself is not going to function. Okay, moving on. Inner ear. Here is where things start to get confusing, and we get lots of terminology that kind of overlaps with each other. So in the inner ear, we're inside the temporal bone, inside that petrous portion of the temporal bone. We're going to have structures made out of bone called bony labyrinth. Inside the bony labyrinth, we're going to have soft tissue. That soft tissue is called membranous labyrinth. There is fluid inside the membranous labyrinth. The tissue of the membranous labyrinth is made out of epithelial tissue, and in certain regions of the membranous labyrinth, we're going to have sensory epithelium. That means that we're going to have sensory receptors inside this epithelium. And these sensory receptors are what is responsible for our sense of balance and our sense of hearing. So let's go to the image. So here we have the inner ear. This area here is called cochlea, like concha. Over here, we have what's called the vestibule, which is this area right here, and the semicircular canals. So the hard bony part is called bony labyrinth. So that would make up, in this region, these would be the semicircular canals. Notice this is different than the semicircular ducts. The semicircular ducts are the soft tissue inside the semicircular canals. So semicircular canals is bony labyrinth. Semicircular ducts is membranous labyrinth. Inside that membranous labyrinth, you will have fluid. So if you take a look at this cross section right here, right out here is temporal bone. In here, we have bony labyrinth. Here we have our membranous labyrinth. Inside the membranous labyrinth, we have a fluid called endolymph. So endolymph is inside the membranous labyrinth. Out here, we have fluid outside the membranous labyrinth. Here it's called perilymph, meaning fluid on the outside. So you can see the terminology is starting to get kind of confusing, and unfortunately it only gets worse. So out here in the semicircular canals, we're surrounded by the temporal bone. Then we have more bone, which is bony labyrinth. Then we have the membranous labyrinth, which has made out of a soft tissue. Inside the membranous labyrinth, we have endolymph. In the bony labyrinth, but surrounding the membranous labyrinth, we have a fluid called perilymph. Now, if we look down here in the cochlea, we can see the same cross section. Out here, we have temporal bone, then we have bony labyrinth. Then we have this structure here called the cochlear duct. So the cochlear duct is also made out of membranous labyrinth, and it is filled with endolymph. Outside of the membranous labyrinth, we will have more fluid, and that fluid is called perilymph. So the same basic structures are here in the cochlea as in the semicircular canals and ducts, but it has a slightly different configuration or slightly different shape. Now getting back to this big picture here, the stuff in yellow, that's bone, that's bony labyrinth. The stuff in, what is that, pink or kind of purple, this is membranous labyrinth. And it's important to point out that the membranous labyrinth of the semicircular canals and the membranous labyrinth of the vestibule and the membranous labyrinth of the cochlea are all interconnected. So these things are all continuous, or they're all connected with one another. So all this structure in here in purple, what's inside of it? Endolymph. Out here, you're going to have perilymph in this shape. In the cochlea, you're going to have perilymph in these two structures, which we'll look at in a little bit more detail here in a second. Okay, so I've gone over the picture. Now let's review the words. Right, the space between the walls of the bony and membranous labyrinth is filled with perilymph, helps protect and support the membranous labyrinth. Inside the membranous labyrinth, you have endolymph. The bony labyrinth is partitioned into three regions: vestibule, semicircular canals, and cochlea. So we saw all three of those. Sometimes you group the vestibule and the semicircular canals together, and when you do that, it's called vestibular complex. So you can either call it the cochlea and vestibular complex, or the cochlea, semicircular canals, and vestibule. Now, if you look inside the vestibule, right, in here, you're going to find two areas of membranous labyrinth. One is called the utricle. Another one down here is called the saccule. So inside the vestibule, you'll have the utricle and the saccule. Membranous labyrinth. So if we look over at the cochlea, you will have the cochlear duct. Going back to the picture, there's a cochlear duct made of membranous labyrinth. Out here in the semicircular canals, you have the semicircular ducts, membranous labyrinth inside the semicircular canal. All right, so that covers these structures. Now things are going to get a little bit confusing. So what we're going to see in the inside the vestibule, inside the semicircular canals, and inside the cochlea, we're going to see a basic setup. You're going to see hair cells that stick into something. When these hair cells are bent, it's going to generate an axis potential. If these hair cells bend inside the vestibule, you'll perceive that as a position change in your head. If hair cells bend over in the semicircular canals, you will perceive this as a rotational acceleration of your head. If these hair cells bend over in your cochlea, you will perceive that as sound. So the basic mechanism for equilibrium and sound are exactly the same. So I just want you to realize you're going to see the same basic setup repeated 3 times. So we're going to go over the first one here. So we're going to be talking about the macula. The macula, going back to this picture, the macula is found in this area here. Inside the vestibule, it is made up of the utricle and the saccule. So utricle and saccule are membranous labyrinth. In the membranous labyrinth, we're going to have a sensory epithelium, just an area that contains sensory receptors. In the utricle and saccule, that membranous labyrinth, in the utricle and the saccule, this sensory epithelium is called macula. So let me repeat that again. In the vestibule, in the utricle and the saccule, we're going to have a sensory epithelium. That sensory epithelium is called macula. So starting from there, let's just read through these terms so we don't get confused. The macula of the vestibule are located on the interior walls of the saccule and utricle. So here we're going to find hair cells that stick into stuff. Right, hair cells are sensory receptors for both equilibrium and hearing. Right, on their apical surface, because remember this is an epithelium, we're going to get 50 stiff microvilli called stereocilia, and one really long one called a kinocilium. So all these hairs are going to project up into a structure we're going to take a look at in a second. When these hairs bend, you will perceive that as a change in head position. So where are we? We're down here inside the membranous labyrinth of the vestibule. Specifically, we're looking at these two regions here called the utricle and the saccule. Here is our sensory epithelium. The name of the sensory epithelium in the utricle and the saccule is called macula. In the macula, we have hair cells with their single long kinocilium and 50 or so stereocilia. And here they're projecting into stuff. The stuff here is called otolithic membrane. So ota or otic refers to ear. Lithic means stone. So this literally means area of the ear stones. And if you take a look in this gelatin-like material, you'll find these crystals called otoliths. So here we have the hair cells with hairs that stick into stuff. In this case, down here in the vestibule, hair cells are in the macula, and those hair cells are sticking into the otolithic membrane. So the job of the utricle and the saccule, the job of the macula, is to detect position of your head. When your head turns to one side, that otolithic membrane moves. That causes the hair cells to bend. That's going to generate an action potential to tell you where your head is. So that's what all this terminology is talking about. Otoliths of the ear stones stuck in the membrane. Let me just cut to the picture. So when your head is normal and upright, this area, this sensory epithelium, is inside the vestibule. Here we have hair cells sticking in the otolithic membrane. Change head position. This gooey otolithic membrane sloshes to one side. That causes all these hair cells to bend over. When the hair cells bend over, it generates an action potential, and that will travel down the axon of these neurons, go to your brain, and tell you that your head is now tilted to one side. And that's just what the rest of these words are telling you. Okay, so just as a review, here in the vestibule, we have two regions of membranous labyrinth, the utricle and the saccule. They contain a sensory epithelium that's called macula. The hair cells in the macula stick in the otolithic membrane. The otolithic membrane moves, hair cells bend. You perceive a change in head position. Now, moving on to our next system for equilibrium. And this involves the semicircular canals. So the semicircular canals, the semicircular ducts, are connected to the utricle and the saccule. We have three semicircular canals: anterior, posterior, and lateral. So what these do is when we move our head in any of these three directions, right, we're going to sense that in the semicircular canals. So at the base of the semicircular canals, we're going to have structures called ampulla. So we take a look at the picture. What's this pink thing? It's all membranous labyrinth. That's the cochlea. That's the vestibule. These are the semicircular ducts. At the openings of the semicircular ducts, we have these structures here called ampulla. So in the ampulla, we're going to have another sensory epithelium that has hair cells that stick into stuff. Those hair cells are going to bend over, generate an action potential, and will perceive a change in equilibrium. So here in the ampulla of the semicircular ducts, we have the structure here called the Crista Ampullaris. So the Crista Ampullaris is the name of our sensory epithelium. If you're in the utricle and the saccule, it's called the macula. Here in the ampulla, it's called the Crista Ampullaris, which contains our sensory epithelium, which is made up of hair cells, which stick into stuff. Here, the stuff that it sticks into is a gelatinous material called cupola. So the cupola is this big dome-like structure which sits inside the ampulla. Now, when you rotate your head, the material, the endolymph inside the semicircular ducts will push, shown right here, will push on the cupola. When the cupola bends over, the hair cells attached to it bend over. That generates an action potential, which travels up our vestibular nerve to our brain and let us know that we've had a position in head change. So the same basic process is going on. hair cells bend, generates an action potential, and we have a change in equilibrium. So just to go over the terminology, right, the ampulla contains sensory epithelium called the Crista Ampullaris. Hair cells stick into the cupula, right? When the cupula is pushed on by the endolymph because we've turned our heads, we perceive that as a position in head change. Okay. Now, the vestibular sensation pathway, this terminology in this chapter has already gotten so complicated. I'm just going to skip this because we have to cut something out somewhere. We just have so much going on, and teaching online is pretty difficult. So if you want to read this and check it out, great. Am I going to test you on it? No. Okay, our final section, let's study the structures for hearing. All right, so hearing is going to be contained inside the cochlea. So that's bony labyrinth. Inside that area, you're going to have the cochlear duct, which is made of membranous labyrinth. Now, we're going to have two regions that can contain perilymph. One is called the scala vestibuli, and the other is scala tympani. So let's go to the picture. Here is the cochlea, cross-section of the cochlea. If we look at one area, one box, we can see our cochlear duct made of membranous labyrinth. Down in here, we're going to have our sensory epithelium, which contains hair cells which stick into something. And when they bend over, they're going to give us a perception or sensation of hearing. For right now, let's look at our larger structures. Here is the bony labyrinth of the cochlea. These are fluid-filled spaces filled with perilymph. The one here on top is called the scala vestibuli. The one on bottom is the scala tympani. If it helps you remember, you can think of, if you're aware, a timpani is a kind of really big kettle drum, and it's kind of low and a bass sound. It's kind of on bottom. That helps you remember? Great. Down here, just underneath, just inferior to the cochlear duct, we have what's called the spiral organ. And the spiral organ is actually going to contain our sensory epithelium. So if we look at that, we can see hair cells with their stereocilia and their kinocilia sticking into something. Here, the structure is called Tectorial Membrane. So when the tectorial membrane moves, it will push and pull and cause the hair cells to bend. And when they bend, we will perceive a sound. So the mechanism is the same as equilibrium, but the end result, our conscious awareness, is something very, very different. All right, I should point out before moving on a couple of other structures. This structure right here is called the vestibular membrane, and the vestibular membrane separates the scala vestibuli with the cochlear duct. And it's kind of distinct because it has this thin structure to it. Down here, we have the basilar membrane, or the basilar membrane, which will separate the cochlear duct from the scala tympani. And this is readily apparent because you have this big, thick structure here next to the spiral organ. So I should also point out down here in the spiral organ, our tectorial membrane, now we have outer hair cells, and we actually have a few inner hair cells, and we have some supporting cells just to support and nourish these cells. Taking a close-up look of the spiral organ. So in here, this is cochlear duct. This is going to be filled with endolymph. This is the basilar membrane. So down here is the scala tympani filled with perilymph. Here we're going to have our outer hair cells, our inner hair cells. You can just see the hair sticking into the tectorial membrane. So again, that membrane moves. That's going to cause hair cells to bend, and we will perceive sound. So the reason that the spiral organ is called the spiral organ, let me go back to the picture. As you can see, it spirals and around and around and around the cochlea. So the cochlea is like a snail shell. You can unwind the whole thing and then wind it back up, and it spins up like a top. So you can see the cochlear duct winding around inside the cochlea, and the spiral organ just winds around with it. That's why it's called spiral organ. So the spiral organ is inside the cochlear duct, and it can detect the movement of endolymph, hair cells bend, and we perceive sound. What do the hair cells stick into? Something called tectorial membrane. So if you want to know head position using the vestibule, your sensory epithelium is called macula. Those hair cells stick into the otolithic membrane. If you want to know rotational information, those are at the base of the semicircular canals. The sensory epithelium is called Crista Ampullaris, part of the entire structure called the ampulla. There, hair cells stick into the cupola. Down here in the spiral organ, which is inside the cochlea, the sensory epithelium is called spiral organ, and the hair cells stick into tectorial membrane. Okay, so this is the last slide. This is not a class in physiology, but I at least want to walk through how exactly the ear works. So sound out in the environment is just waves of air molecules bumping into each other. Just like you throw a stone in the water, waves ripple out from it. If you make a loud sound, air molecules start bumping into each other and you get compression waves. Those compression waves of air molecules hit your tympanic membrane. Tympanic membrane starts vibrating. That causes your auditory ossicles to vibrate. Your auditory ossicles will actually magnify that intensity by a factor of 20 or even as much as 30. So that energy is deposited in the round window. Now the round window, that energy is deposited in the oval window. The oval window is connected to the cochlea. Specifically, it's connected to the scala vestibuli, right? That space filled with perilymph. So those energy waves, those sound waves, are going to be transported into the perilymph of the scala vestibuli, and they're going to travel some distance. Now, when they travel a certain distance, and I'll explain to that in a second, the energy will be deposited. It will move into the cochlear duct, where it will vibrate endolymph. The vibrating endolymph will cause the tectorial membrane to vibrate, and when the tectorial membrane vibrates, it causes hair cells to bend over. That energy then continues into the scala tympani, and then the scala tympani, that is then connected to a structure called the round window, where you have a little elastic membrane that allows to go in and out. So let me break this down a little bit more. Vibrating air molecules cause the tympanic membrane to vibrate, which causes your ear bones to vibrate. That magnifies the sound by a factor of 20. That energy enters the scala vestibuli. It travels a certain distance, and then it is transferred to the cochlear duct, where you get vibration of the tectorial membrane, which causes hair cells to bend over. You perceive a sound, and then energy continues and flows through the scala tympani and is released through the round window. So think of these ducts as a garden hose, right? If you put a cap at the end of a garden hose and try to add more water to it, nothing's going to happen. If, however, you put a balloon at the end of the garden hose and then you turn the spigot on, water will rush into the hose because the balloon can expand. So that balloon at the end of the garden hose, that's the round window. So every time the stapes pushes on the oval window, it displaces perilymph in the vestibuli, and then the oval window pushes out. So every time the oval window pushes in, the round window pushes out, and that allows for the vibration of the fluid in the ear. Now you might be asking yourself, well, if all that's going on are hair cells bending over, how do the hair cells know what tone, right? How can you tell pitch, right? How is that possible? And that actually has to do with a property of sound itself. So ask yourself for a second, what type of sound travels the farthest? A high-pitched sound or a low-pitched sound? Most people tend to think that a high-pitched sound will go the farthest because, high-pitched sounds are very annoying. But if anyone has been outside of a club or something like that, what do you hear outside? You hear thump, thump, thump. You hear the bass line. Low frequency sound travels much farther than high frequency. So the way you're able to determine pitch is dependent on which hair cells bend over. So if you look here in the cochlear duct along the spiral organ, the hair cells way down here at the end, right, when these hair cells bend over, they will generate a sensation that you will perceive as a low pitch because the energy waves travel very deep into the cochlea. If, however, you have a very high frequency pitch, they will only travel a short way into the cochlea and activate the hair cells right here. If these hair cells are activated, you will perceive a high pitch noise. So, depending on the wavelength of the sound, if it's low, it will penetrate down deep into the cochlea down here, and then you'll perceive a low pitch noise. If the frequency is very short, it only travels a short distance, these hair cells here will be activated and you will perceive a high pitch noise. So again, this is not a physiology class, but it's only one extra step to explain how actually our sense of hearing works. So now maybe it makes a little bit more sense. How can your ability to tell whether or not you're standing on your head with your eyes closed is directly related to your ability to hear things? Well, hopefully now that all makes sense to you. So again, I apologize. The material in this chapter is very confusing. Lots of overlapping terms, perilymph and endolymph and macula, Crista Ampullaris and spiral organ. A lot of the stuff all kind of runs together. So I strongly recommend read the book and actually write out the exact definitions of what's the difference between a semicircular canal and a semicircular duct. What are the different types of sensory epithelium, right? Try to get all that stuff really clear in your head because a lot of people get confused in that terminology. Right, with that, we are done with special senses. Wish you guys be safe, take care of yourselves, and we'll see you next time. Take care.